Good morning, everyone. In today's session, we will try to understand the concepts of the biodegradation and the role of microorganisms, emphasizing on genetically engineered microorganisms in the process of biodegradation. Biodegradation is defined as the biologically catalyzed reduction in complexity of chemical compounds. That is, it is a biologically catalyzed reduction process, breaking down into smaller compounds. The complexity of the chemical compounds is broken down into smaller compounds. That means it is a process by which organic substances they are broken into or broken down into smaller compounds by living microbial organisms. That means the microorganisms break the organic substances into smaller compounds. So this is essentially the definition of biodegradation. When the process of biodegradation is complete, it is called as mineralization. However, in most cases, the term biodegradation is generally used to describe almost any biologically mediated change that is observed or seen in a substrate. Right? So, when the biodegradation process is completed, now this process is called mineralization. Now, what is biodegradation? It is a biologically catalyzed, that is, through living microbial organisms, the complex organic substances are broken down into smaller compounds. In most of the cases, the biodegradation or the term biodegradation is usually associated with almost any biologically mediated changes that happen or occur in a substrate. So, understanding the process of biodegradation requires an understanding of the microorganisms that make the process to work. So, it is not only about understanding what is biodegradation, it is equally important to understand about microorganisms that make the process of biodegradation to work. So the microbial organisms, we know that they break down or transform the substrate or the organic substances through metabolic or enzymatic processes. So this process, that is through metabolic or enzymatic processes, is divided into two types of process. Number one, the growth, and number two, co-metabolism. Okay, so the metabolic or enzymatic process by which the microbial organisms transform the substance is divided into, or it is based on two processes, that is the growth and the co-metabolism. Now in growth, an organic pollutant is used as a sole source for carbon and energy. Right? So what happens in the process 1, that is growth? An organic pollutant is used as sole source of carbon and energy. Now, what is happening in process 2? It is defined as the metabolism of an organic compound in the presence of a growth substrate. Now, this is used as the primary carbon and energy source. Okay, so in process 1, that is growth, an organic pollutant is used as a sole source of carbon and energy. Now, this process results in complete degradation, that is mineralization, as said in the previous slide. Degradation or mineralization of organic pollutants. Now, what happens in the process 2, that is co-metabolism? This is the metabolism of an organic compound in the presence of a growth substrate, which is used as a primary carbon and energy source. Now, there are several microorganisms which include fungi, bacteria, and yeast, and are known to be involved in the process of biodegradation. And algae and protozoa, there are reports about algae and protozoa, but the reports are very less. That means the reports are scanty regarding the involvement of algae and protozoans used in the process of biodegradation. So it is most of the time the microorganisms which include are the fungi, bacteria, and yeast. Okay. Biodegradation process. So this process vary greatly, but frequently the final product of the degradation is carbon dioxide. So the organic material can be degraded aerobically with or in the presence of oxygen, or anaerobically in the absence of oxygen. So the process of biodegradation, even if it varies significantly, most of the time the final product of the degradation is carbon dioxide, and the organic matter can be degraded both aerobically and anaerobically. Right. Now, what is biodegradable matter? Which matter to be considered as biodegradable? Biodegradable matter is generally an organic material such as from the plant and animal matter, and could be from other substances originating from living organisms, or artificial materials, say for example oil, PCBs, that is polychlorinated biphenyls, PAH, that is polyaromatic hydrocarbons, radionuclides, and metals. So all these constitute to be called as biodegradable matter. So it is generally the organic material such as plant and animal matter, and other substances originating from the living organisms, and most important is artificial materials. So these artificial materials include polychlorinated biphenyls, polyaromatic hydrocarbons, radionuclides, and metals. Right? So the term biodegradation is often used in relation to the ecology, in relation to the waste management, and mostly associated with environmental remediation. So we call it as, or we know the process as, bioremediation. Right. So this biodegradation, it is often associated with respect to the relationship to ecology, waste management, and environmental remediation, which we know as bioremediation. Now, what is bioremediation? It is synonymous with the process of biodegradation. Now, bioremediation process can be divided into three phases or levels. Right? Number one, natural attenuation. Number two, biostimulation. Number three, bioaugmentation. So the process of bioremediation can be divided into three phases or three levels. Number one, natural attenuation, biostimulation. Number three, bioaugmentation. So what happens in natural attenuation? First, through natural attenuation, the contaminants are reduced by the native microorganisms without any human augmentation or without any involvement of human activity. That is called as natural attenuation. So we are speaking about three different levels of the process of bioremediation, which is an associated process of the term biodegradation. So bioremediation is divided into three phases or levels, and the first phase is natural attenuation, and we know that the contaminants, they are reduced by the native organisms or the microorganisms, and there is no involvement of or augmentation by human activity. Number two, the second one, biostimulation. Biostimulation is employed where nutrients and oxygen are applied to the systems to improve their effectiveness and to accelerate the process of biodegradation. Right. So bioremediation can happen in three different phases. So first phase is natural attenuation, nothing to do with human activity. It is all about the reduction of the contaminants, the process of reduction of the contaminants by native microorganisms. And in biostimulation, there is involvement of the human activity. You are adding the nutrients and oxygen. Microorganisms will be there. You are adding nutrient and oxygen so as to improve the effectiveness, the microorganisms effectiveness, and to accelerate the process of biodegradation. Now what happens in bioaugmentation? During bioaugmentation, microorganisms are added to the system. So natural attenuation is the involvement of native microorganisms. Number two, biostimulation where nutrients are added, and bioaugmentation where microorganisms are added. Right? And many factors influence the microorganisms to use pollutants, the organic pollutants as substrates or to co-metabolize. Now, what are these factors? The factors could be like genetic potential of the microorganisms and certain environmental factors such as the temperature, the pH, and the available nitrogen and phosphorus as sources. So these seem to determine the rate and the extent of degradation. So these factors are very much critical to determine the rate and the extent of degradation. Number one, the genetic potential of the microorganisms, and environmental factors such as the temperature and pH and the availability of nitrogen and phosphorus sources. So all these factors put together, they determine the rate and extent of degradation. Thus, applications of genetically engineered microorganisms, GEM, so abbreviated as GEMS, genetically engineered microorganisms, in the process of bioremediation, it has received a great attention or great deal of attention. Why? Because you have a possibility to modify the genetic potential of the microorganisms, to alter the environmental factors that is required by the microorganisms, such as you can always alter the temperature suitable for the microorganisms, you can alter the pH, and you can also vary the nitrogen and phosphorus sources. Since you can manipulate all these factors which influence to degrade the organic pollutants, so it is possible with the help of GEM, that is genetically modified or engineered microorganisms. Thus, bioremediation has received a great deal of attention. Now, these genetically engineered microorganisms have higher degradative capacity because you have modified them, you have engineered them to suit the needs. So, they have higher degradative capacity and have been demonstrated in many cases successfully for the degradation of several or different pollutants or various pollutants under defined or specific experimental conditions. But the limitation is the ecological and environmental concerns, because these are genetically engineered microorganisms. No doubt they have the higher degradative capacity and have been demonstrated successfully for the degradation of various pollutants under defined conditions. But the concern is about the ecology and the environment and the regulatory constraints that are in place. So these are the major obstacles for the use or for testing or for exploiting genetically engineered microorganisms at or in the field levels. Right? So these are the important obstacles. Number one, the ecological concern. Number two, the environmental concern and the regulatory constraints. So these are the major obstacles for the use or for testing of genetically engineered microorganisms at the large scale or in the field. Right? So now this illustration gives you an idea about the factors that influence the process of bioremediation. So this is bioremediation of pollutants utilizing biodegradation abilities of microorganisms. We have seen the three different levels: the natural attenuation, then we have the biostimulation, then we have bioaugmentation. So the factors that affect are the abiotic factors, and we have the biotic factors. Under abiotic factors are the pH, temperature, moisture, and aeration. Under biotic factors, we have the competition and then the antagonistic interaction. So these are some of the important factors that are responsible, and if the genetically engineered microorganisms can be manipulated in such a way that they possess higher degradative capacity, right? So now there are some biodegradable pollutants. We'll see about each of these pollutants. Number one are the fuels, the hydrocarbons, and then we have polychlorinated biphenyls, polycyclic aromatic hydrocarbons, pesticides, and dyes, and some other synthetic, that is artificial chemicals like radionuclides and metals. So these are extremely resistant to the process of biodegradation by native flora compared with the naturally occurring organic compounds, so that are readily degraded upon introduction into the environment. So they are resistant to the native flora, so we have to go for the genetically engineered microorganisms. Then we have the hydrocarbons. So we'll try to understand about each of them a little more. Hydrocarbons, they are the organic compounds whose structures consist of hydrogen and carbon. And these hydrocarbons, they are seen as linear or branched or cyclic molecules. They are observed as aromatic and aliphatic hydrocarbons, and the first one is has the benzene ring in its structure. The aliphatic ones is seen in the forms alkanes, alkenes, and alkynes. And then we have the polycyclic aromatic hydrocarbons. So they are the most important pollutant, and they are widely found in air, soil, and sediments. And the major source of polycyclic aromatic hydrocarbon pollution is the industrial sector, the industrial productions. So these polycyclic aromatic hydrocarbons, they can easily enter into the soils. That is, they can easily sorb into organic rich soils and sediments and accumulate even in fish and other aquatic organisms, and mainly, and they may be transferred to humans through the consumption of these aquatic animals, mostly the seafood. So the biodegradation of polycyclic aromatic hydrocarbons, they can be considered as on one hand to be part of the normal process of the carbon cycle, and on the other as the removal of man-made pollutants from the environment. So the use of microorganisms for the process of biodegradation of polycyclic aromatic hydrocarbons or these pH contaminated environments seems to be an effective technology for restoration of the pollution sites or polluted sites. Then we have polychlorinated biphenyls. These are the mixtures of synthetic organic chemicals, that is PCBs, and due to their non-flammability and chemical stability, and also high boiling point and electrical insulating properties. See, the properties which the PCBs possess: non-flammability, chemical stability, high boiling point, and electrical insulating property. So because of these, PCBs are used in hundreds of industrial and as well as other commercial applications. So consequently, the PCBs are toxic compounds that could act as an endocrine disruptors and cause the cancer, since it is having a wider applications in several industry and in sometimes in even in some commercial applications. So it is widely used, but the thing is it act as an endocrine disruptor and causes cancer. So therefore environmental pollution with PCBs is of great concern, or the most interesting concept to be taken under bioremediation. Then we have the pesticides. These pesticides are substances or the mixture of substances which are intended for preventing, destroying, repelling, or mitigating any pest. So, from agricultural point of view. So these pesticides which are rapidly degraded are called non-persistent, while those which resist degradation are termed persistent. So there are two categories of pesticides: the non-persistent pesticides and the persistent pesticides. So pesticides which are rapidly degraded are called non-persistent pesticide, and the pesticides which resist the process of degradation are termed persistent pesticides. So the most common type of degradation is carried out in the soil by microorganisms, especially the fungi and bacteria that use pesticides as the source of food. And now we have the dyes. These dyes, as we know, they are widely used in the textile industry, the rubber industry, and even in paper industry, printing industry, and in color photography, pharmaceuticals, cosmetics, most importantly, and many other industries. So the azo dyes, so which are aromatic compounds, are the most important and the largest class of the synthetic dyes which are used in commercial applications. So azo dyes are aromatic compounds, and we have seen the industrial segment, that is textile, rubber, paper, printing, photography, pharmaceuticals, cosmetics, and so on and so forth. So these dyes are poorly biodegradable because of their structures, and treatment of wastewater containing dyes usually involves physical and or chemical methods such as adsorption, coagulation, flocculation, oxidation, filtration, and electrochemical methods. So these are another important class of the biodegradable pollutants which are of great concern under bioremediation. Then we have the synthetic chemicals, the radionuclides. Is these radionuclides is an atom with an unstable nucleus characterized by excess energy available to be imparted either to a newly created radiation particle within the nucleus or via internal conversion. So during the process, what happens during this process? The radionuclide is said to undergo radioactive decay, resulting in the emission of gamma rays and or subatomic particles such as alpha particles or beta particles. So again, these are the synthetic chemicals which contribute to the or which are listed under biodegradable pollutants. And we have heavy metals. Unlike organic compounds, these metals cannot be destroyed, but they must either be converted to a stable form or it has to be removed. So they cannot be destroyed. They must either be converted into a stable form or have to be removed. So bioremediation of metals is achieved through biotransformation. So it is always the transformation. So that is conversion of the heavy metal into a stable form, right? So this is an illustration about the involvement of the microorganisms in the process of bioleaching, biosorption, biomineralization, and the transformation. So essentially, the mechanisms by which microorganisms act on heavy metals include biosorption. So biosorption is the process by which the microorganisms degrade or act on heavy metals. That is metal sorption to cell surface by physicochemical mechanisms. Then we have bioleaching, where heavy metal mobilization occurs through the excretion of organic acids or methylation reactions. And next we have biomineralization, where heavy metal immobilization through the formation of insoluble sulfides or polymeric complexes. And then it is about transformation, that is intracellular accumulation and enzyme catalyzed transformation, that is redox reactions. So I have picked four important ways or mechanism by which microorganism or the microbial cells act upon. That is number one, the biosorption, where the metal sorption to cell surface by physicochemical mechanism occur, and then bioleaching, where heavy metal mobilization through the excretion of organic acids or methylation reactions occur, and then biomineralization, where heavy metal immobilization through the formation of insoluble sulfides or polymeric complexes occur, and then we have intracellular accumulation and enzyme catalyzed transformation, that is redox reactions. So we have tried to understand what are the or what is biodegradation, and what is bioremediation, and what are the different levels of or phases of bioremediation, that is natural attenuation, biostimulation, bioaugmentation, and what are the different categories of or the various organic pollutants, and how the microbial cell or the mechanism by which the microorganisms act upon these heavy metals. And there are four processes: biosorption, bioleaching, biomineralization, and transformation. So I'll stop my discussion over here. We can continue our discussion in the next session. Thank you very much.